In this video, we'd like to take on the second of our two chapter goals, that is predicting reaction direction. This is a skill that we need to develop because we're going to see dozens, if not hundreds, of reactions in the next 5 or 6 weeks. And we want to get a sense of whether they're moving in the forward direction or the backward direction and which way they have a tendency to go. And this is going to be simple in some cases and much more complex in others. Let's start this process by looking at two fairly simple reactions that we want to talk about. These are involving molecules we've seen before. Here's a reaction, a reversible reaction, and here's another reversible reaction. I'd like you to take a moment, pause the video, and name all of the compounds that are present in these two reactions. We've seen them all before. The second thing I'd like you to do is determine if there's anything missing. In fact, each reaction is missing something on the left side or the right side, and I'd like you to figure out what it is. And lastly, I'd like you to tell me what linkages are involved here. Up in this reaction, we are breaking a linkage, and in this reaction, we're creating a linkage. Let's see if you can remember those from previous chapters. Pause, take care of these questions, and we'll return. Okay, let's take these apart. First of all, this is a nucleotide. We call it ATP. We already mentioned that this is the energy currency of cells, but it's a standard ribonucleotide with a base, a sugar, ribose, and a 5' triphosphate. Over here, we have ADP because we've split off a phosphate. Now, this is a phosphate down below, but we sometimes abbreviate that P subscript I for inorganic phosphate. We're going to use that an awful lot, so let's leave it at that. Down here, you may recognize D-glucose. That is a aldohexose with 6 carbons. We also have a PI, and if we take a PI and connect it to glucose on the 6th position, very simply, we get glucose 6-phosphate. All of these things we've seen before. Let's go to the linkages, the last question at this point, because that's a little easier. When we break that linkage right there, do you know what that is? We've talked about it once or twice before. It's going to become very important going forward. Whenever you have two phosphates that are linked together, you have a phosphoanhydride. Then in the other reaction down here, we're taking a phosphate and jamming it onto an alcohol. And when we do so, we have a phosphoester. We've seen that many times before. And again, this will play an outsized role moving forward. These are different linkages, but they both involve something that's missing. Whenever you take a phosphoanhydride, for instance, and break it and chop a phosphate off, you need to have an extra species, and that is going to be water. So that's needed to balance. And that's true down here as well. If we take a phosphate and put it on an alcohol, we've lost an oxygen here, and that means we are going to generate a extra water. And so that's an important part of both of these reactions to balance them. So at this point, we have these two reactions, and the next question I would like to ask you is just a thought question, is which way do these reactions tend to favor? The forward direction, the reverse direction, or are they evenly favoring the forward and the reverse direction? Well, how would you know? You'd really have no idea other than intuition, but here's what we can do. We can think about it in two different ways. The first is we can write out reaction energy diagrams. And if we do that with this particular first reaction, the splitting of ATP into ADP and phosphate, we would say that we have ATP here, and water, of course, and over here we have ADP and phosphate. We've got the substrate on the left and the products on the right. And if we look at this reaction energy diagram, we see two things. One is there's a big hump in the middle. That slows this thing down so that it doesn't occur very rapidly at all. However, we also see that the ATP over here is higher in energy than the ADP in phosphate. And that's the more critical part that we're interested in now. This difference right here, which is going downhill, tells us that the conversion of ATP to ADP in phosphate is going to release energy as we go from substrate to product. And because it releases free energy, it has an overall favorability in the forward direction. And it's also pretty big in its elevation change there. It's a fairly large drop of energy. That means it's going to be even more favorable than others that have a smaller drop. Now, if we go down to the bottom here, we can see a different scenario. In this case, we have a situation where the conversion of glucose to glucose 6-phosphate, that is glucose plus Pi goes to G6P, that's an uphill reaction. And that means we have to put energy in. And what that means is going to favor the reverse direction. It's going to want to flow in this way. Now, you got to understand this is kind of like the stability of these molecules, and it's thought of in a potential energy sense. ATP has more potential energy than the combination of ADP and phosphate. So, just like water at the top of a waterfall, it's going to tend to flow in the forward direction towards ADP and phosphate. It's the opposite for the reaction of glucose and phosphate to make glucose 6-phosphate. It's going to inherently favor going in the backward direction. Well, how do we know these things? Well, it has to do with how we measure what this is. And this is going to be called the delta G naught prime. You're not ever going to have to do this calculation, but it is an important idea. If we take a standard concentration of ATP, ADP, and phosphate, if we take 1 molar of each of those, put them in a reaction, and let them go to equilibrium, we would find something quite astonishing. We would have 420 of ADP and 420 of phosphate for every 1 of the ATP reactant that we have. If we throw in the temperature and a constant and a negative charge, we can't forget that negative charge, we can calculate the delta G naught prime. And that's what this is up here. It's the change in free energy from substrates to product. And in this case, the delta G naught prime is negative 30 kilojoules per mole, approximately. And what that means is the negative means it's downhill. It favors the forward direction in general. And the 30 means it's a pretty big downhill. It's a large change in free energy that pushes us forward. That's for this reaction up there. The delta G naught prime for the second reaction, if we did the calculation, is going to favor the reverse reaction, and therefore it's an uphill delta G. And it turns out, I think it's about positive 14 kilojoules per mole. The positive means it's uphill and therefore favors going backwards towards the substrates, and the 14 means it's about half as much so compared to the ATP. So we get direction and we get the intensity of preference for forward or backwards from this delta G naught prime number. It's very important. The units are kilojoules, which are energy, for every mole of the substrates and products that we use. So delta G naught prime is an important principle for determining which way the reaction is going to go, which, like we said, is our goal in this chapter. We'll pick it up on the next video with a follow-up.